The topic for today's lab is on blood grouping and typing and cross matching. So if everybody can mute yourself. Now, what is blood typing? Well, let's go back to the red blood cell and ask ourselves a couple of questions. In the lecture, we learned, can you mute yourselves? I'm echoing in somebody's speaker system. Please mute yourself. We learned in the lecture that the red blood cells do not have nuclei, do not have mitochondria. And here's the third thing that red blood cells do not have. Guess what? From your ANP1 lecture, we said all cell membranes carry identifier proteins. These identifier proteins are often referred to as PIPs, P-I-P-S. PIPS, what does PIPS stand for? It stands for personal identifier protein. Now, personal identifier protein is called as antigens. So PIP, personal identifier proteins. All antigens are proteins. We'll talk more about it when we come to the immune system. For now, so what are these identifier proteins? They are unique to each of you. Each person has their unique, just like your DNA is determined by your DNA. All cells in your body have identifiers with the exception of red cells. Your red cells has no personal ID, meaning if I remove a couple of red cells from 6 different people and throw them around, I can never prove who it belongs to. There is no identity of the owner. However, what they don't have in personal ID, they have what is known as group ID. And this group ID is based on a protein found on the surface of red cells. So the early workers discovered that the entire world population has 4 possibilities. There are only 2 proteins that can be found on the surface. They call the first one the A protein. A protein. So what is A? The A protein is also referred to as the A antigen. Now, the definition of antigen will cover when we come to the immune system. For now, just remember that the surface identifier protein in red cells could be a protein referred to as A, and then they found some people had a B protein, and some people had both, and there were a group of people who had no protein at all. So if they had no protein, we call them zero, so they call them O. This is the beginning of what is known as blood grouping. This is the oldest and the most commonly used blood grouping technique around the world for the entire world population. So the entire world, the people in the world have to belong to one of these 4 groups. So if you are born with a genetic gene to have the A protein, we call you blood type A. If you're born with B, we call you B type. If you have both, we call you AB. If you have none, we call you O. So what is the criteria for blood grouping? The presence of the antigen. So it is the surface protein on the cell membrane of a red cell that determines your blood type. This mechanism is the first to recognize your blood type. And like I said, is genetically determined. What I want you to do today, and make a note for yourself, is to open your textbook. And in your textbook, there's a population distribution of the various blood types for the United States based on race and ethnic groups. There are 4 major groups in the United States: Caucasians, African Americans, Native Americans, and Asian Americans. And then you have others. So I want you to go and write down the distribution of blood groupings in these various percentage, the various groupings of ethnic peoples, and you will see an amazing difference. Native Americans are majority different from Caucasians, and Africans Americans are different. And this is applicable to all parts of the world, all countries, all regions. So you can, if you're interested, those of you who are in Africa, you can look up West Africa. In West Africa, you can look up individual countries. You will find the blood group distribution. And even in Africa, the blood groups vary from tribe to tribe. If you're looking at Nigeria, you have three major tribe groups, the Hausas, the Igbos, the Yorubas. You can look up what is the most common group of uh, blood types in the different tribes. So it can vary by tribe, it can vary by region, it can vary by the ethnic groupings. So you can read up all that. Next, the second part of blood typing is the presence of protein in plasma. We refer to them as antibodies. Now, like I said, the detailed concepts of antigens and antibodies will be discussed in the immunology lecture, but for now, just note that antibodies are proteins found in plasma. And the basic premise of immunology is your body protects itself against that which it is not. Repeat, your body's defense is against all foreign proteins, all proteins that is not yourself. So therefore, when we talk about antigens, we talk about self-antigens and non-self antigens. So if you look at this picture, and if you belong to the blood group A, and you have A antigen is referred to as the self antigen. Self, meaning it's you, it belongs to you. So it's referred to as self antigens. And antibodies are proteins against non-self. So what is a non-self antigen? So if you are an A, who are you not? What is non-self to you? B. Am I right? Because you're not B, right? So your body's defense system is designed to protect you against the B protein. Why? Because you are not B. So therefore, in your plasma, look down below here, you will see the protein antibodies are anti-B. So what is the purpose? What do antibodies do? If by accident a B antigen carrying red blood cell comes into your circulation, these antibodies will attack and destroy it. They will cling to the surface and destroy that cell. Why? Because your immune system says, I don't tolerate anything that is not me. I only tolerate me, self. So in blood grouping, you're looking at two aspects. You're looking at what is found in the plasma and what is found on the surface of the red cell. So on the surface is your self antigen, and in the plasma is your non-self antibodies. So this is the basis of the blood grouping. So if you walk down the line here, you look at type B. Type B will have anti-A. Now look at type AB. Hello, they have both A and B antigens. They cannot have any antibodies. So their plasma has no antibodies. Why don't they have antibodies? Because you cannot commit suicide. You cannot self-destruct. You can have a protein that destroys your own body cells. So therefore, people with type A are born with no antibodies in their plasma. Let's go to type O. These ones have no antigens. Zero antigen. Therefore, they protect themselves against both A and B. So the plasma of blood type O has both anti-A and anti-B. So this is the basic premise you need to understand. Don't try to memorize this. Be logical and reason it. So the first logic is ask the question, what antigen do I have on my cell? Second question, what should I have to protect me? What I don't have, anti-B. That's the second. Now, researchers then found out that other than A and B, we had one more protein that was found on the surface. And these surface proteins were referred to as rhesus antigens. That's what this, if you look at it here, RH stands for rhesus. Rhesus factor. I'm going to move this a bit. They found out that the entire world population had two possibilities. Either you had the rhesus or you had no rhesus. So if you had the rhesus factor protein on the surface, we called you rhesus positive. If you are born without it, we said you are negative. And only 2% of the world population is born negative, meaning only 2% of the population has no rhesus factor. 98% of the world population, including possibly you and me, have the rhesus factor. And if you have it, we say you are positive. So what was the historical evidence of this? Well, when they were studying blood cells, they were studying them in primates, monkeys and chimpanzees, and they found out that this rhesus factor was found on the rhesus monkeys of West Africa. You find these monkeys in Africa and in Asia, and they found these proteins on the surface, and therefore, because it originated and found on that, we also found the same rhesus factor in human red cells, so we called it the rhesus factor. So what are the two criteria for blood grouping? Number one, ask the question, what's the self-antigen? Number two, ask what is the antibody in the plasma? And number three, ask the third question, do you have the rhesus factor or do you not have the rhesus factor? And thus, the blood grouping falls into two categories, type A positive, B positive, AB positive, O positive. What does it mean? All of them, in addition to the absence or presence of antigens, carry the rhesus protein. And then you have all the negatives, A negative, B negative, AB negative, and O negative, meaning they do not have any rhesus factor. So again, same thing. What would you find in the plasma of people who are AB, ABO negative? What is the premise of the body's immune system? You protect yourself against what you are not. So because you have the rhesus factor, 98% of the world population, they will not have rhesus antibodies. There are no rhesus antibodies in rhesus positive people. However, rhesus negative people, because they don't have the factor, will have rhesus antibodies. Now, typically, they may not be found in circulation. When I go to the next couple of slides, it plays a critical role on pregnant women. I'll talk about it there. So right now, this is what we have concluded. So the first part is understanding what is blood grouping. Blood group is based on the presence of the antigen and the presence of antibodies, which are the protective. So who makes these antibodies? We'll talk about that later in immunology. So this is just a breakdown of it. The next thing is, what will happen in a situation where a person is exposed to some blood that is not of his type? Well, basically, this is the reason why we started what is known as grouping and cross-matching. I want you to understand this. Blood, as we learned in the lecture, is the essence of your life. And therefore, the loss of blood can lead to the loss of life. Agreed? That's what we heard in the lecture. So man's quest to preserve life in a situation where you lost a lot of blood and death was certain, the only way to preserve life was to reinfuse you back with blood. There is no substitute for blood. You can go and read, there are some recent artificial bloods and you can read all about it. But to date, we have not found an alternate to blood. Blood can only be replaced by blood. And blood, being a liquid connective tissue, became the first tissue transplant ever carried out in humans. Again, interview questions. If I'm interviewing you, I will ask you, what's the first tissue transplant ever undertaken in human beings? The answer is blood. Go back historically. If anybody is interested for your lecture to do a paper on blood grouping and cross matching, that's a good topic. Historically, man's attempt was disastrous. They said, hey, blood is blood, so let's take pig's blood and give it to humans. Let's take monkey's blood and give it to humans. They found everything they did killed the person rather than revive the person. And the only way you can replace blood is through human blood. But then they ran into serious problems because they found out, oh, just because you're a human, it doesn't mean you can take one person's blood and put it into another person because of these antigens and antibodies. So now we came up with an idea of finding a match. So we said, if you have A blood, we have to find you A blood. If you're B, we have got to find you B. If you are AB, we got to find you AB, because otherwise the antibodies present will destroy the transfused red cells. So thus we came about the concept of grouping. So in transfusion, when we talk about transfusion, there are two-step processes. The first process is called as grouping. In grouping, we check the antigen in both the donor and the recipient. And this is especially for the donor. I say, okay, you are the person who needs the blood, you are the person who's willing to give the blood. So first thing I want to check is ask myself, what antigen do you have? Then the second step is I have to look at the recipient's plasma. I have to look at the plasma and say, hey, what type of antibodies do you have? Why? Because I know if I give a person who is A, B blood, watch this here, if B comes into the A blood, the antibodies in the plasma of the person with A will attack and destroy this. Total destruction, defeats and negates the entire purpose of blood transfusion. So once again, remember, when we're doing cross-matching, we take the donor and the recipient cells and find out the group, which group. Second, we take the recipient's plasma and check the antibodies to make sure there is no opposing antibody. And this is what the lab does when you ask for a blood grouping and cross-matching before you transfuse your patient. They take the donor's red cells, dump it in the recipient's plasma, and watch. And what they are watching for is clumping. The clumping of red cells is called as agglutination. When the red cells agglutinate, they are clumping together because the antibodies are sticking to the surface and will eventually destroy the red cells. So they watch for agglutination. And I am going to assign, your first assignment for the lab will be to do an activity of blood typing. It's going to be worth 10 points. It will be released later today, and it will be due next week by the weekend. That's your first assignment for 10 points, blood grouping and cross-matching. And you're going to do this and see the disastrous results you're going to get by mismatching the blood groups. All right. So that's what you're going to do. That's your activity. For the rest of the time, you're going to study this and go over this exercise, which I'm going to go over briefly. And I'm going to ask you questions. Now, this is not supposed to be present. Why this is showing all. So let's see if I can. All right. So here we have a table, and this is found in a lab manual. And we're going to ask the question. What's the question? The question is, if you have blood type A, what antibodies do you expect in your plasma? What do you expect?
B antibodies, isn't it?
B. What type of antigen do you expect in your, on the surface of your red cells? The
It's
answer
the
is
A
right there,
antigens, yeah.
A. So we're going to have A. Now the question becomes, who can you donate to, who can you receive? So A can donate to A, and A can donate to AB. Why is it that they can donate to AB? Remember, in the plasma of a person with AB, they do not have antibodies. So A cell can swim around in the plasma of a AB person with no fear of being attacked because they don't have the antibodies. Now, on the other hand, A can receive only from A. They are compatible, and they can receive from O. How can O be received by A? Because remember, O has no antigens. So even though the person with type A has anti-B has no way to attack because the O blood cells have no antigen. If there's no antigen, there's nothing to attack. So that is the premise. We're not going to go into genotype. Genotype is important when we're talking about different trait. So let's look at blood group B. So what do we know about blood group B? We know that blood group B has antibodies against who? A. What's the antigen on the red cell? B. Who can they donate to?
B and AB.
B and AB. Who can they receive from?
B and O.
Good. Let's go to AB now. What antibodies will AB have in their blood? I said it 30 seconds ago. None. AB has no antibodies. They're carrying both the antigens. What type of antigens do they have on their surface? Both A and B. Who can they donate to? Now watch now very carefully. AB has both antigens, right? Can AB give to A? Yes or no?
No.
No. Can
No,
AB give to
no,
B?
No.
Can AB give
no,
to O?
no,
No.
No. So AB can only give to AB. They cannot give to A because A has anti-B. They cannot give to B. B has anti-A. They cannot give to O. They are both anti-A and B. That will destroy the cell. But who can they receive from?
O.
Everyone?
Oh,
Everybody.
everybody.
Why? This is why AB is called as a universal recipient. AB is a universal recipient because their plasma has no antibodies. A can come in, no problem. B can come in, no problem. AB comes in, no problem. O comes in, no problem, because there are no antibodies in their plasma. So AB is referred to as a universal recipient. That's a common exam question. If I ask you, who is a universal recipient? You say AB, and I ask you for the answer. What is the reason they are universal recipient? The answer is they do not have antibodies. They do not have blood type antibodies. Don't give me any other answer, I'll give you 0. This is the only answer I expect. So let's go to look at blood group O. What antibodies will O have? Both, because they don't have an antigen. What antigen do they have on the red cell? None. They're born without any antigen. Who can they donate to? Tell me why now. What's the reason O is a universal donor? They can donate to A, they can donate to B, AB, and O. Because they do not have antigens, there is no even if the antibody is there, they cannot attack it. So if you give blood group O to a person who's A positive, nothing happens. It is received. Why? Because the red cell has no antigen, the anti-B has no way to attack. So that is why O is referred to as a universal donor. Again, exam question. Who is a universal donor? Blood type O. Why? Because they do not have surface antigens. That's the reason. Don't give me any other answer. But who can O receive from? Unfortunately,
Only O.
O. Only O, because any other blood will be attacked by antibodies because O has anti-A, anti-B. Now the same sequence for A negative. Who can A negative? What do we find in A negative people? You will find B negative antibodies. What's the antigen? A negative. What does the negative sign indicate? Negative means they do not have what? What is it
Rhesus
that they do
factor.
not have?
Rhesus factor.
Exactly. So you can work this in the table. That's your exercise for today. Everything is the same except the negative says in negative people without rhesus factor, everything is the same. So here's my question to you. Can you give, pay attention here now, this is A positive. Can you give A positive blood to a A negative person? Can you?
Yes, but only once.
Absolutely wrong. Your patient will die.
Because it's self antibody.
Because the rhesus factor is foreign to a negative person. The negative person's immune system is going to recognize the factor as a foreign protein and start making antibodies that will attack it. So a negative person cannot receive positive blood. Do not try it. You will kill your patient. Don't do it. However, the reverse is true. You can give B negative or A negative to a A positive person. Why? Somebody explain to me. I can give a negative blood to positives, but I cannot give positive blood to negative. Why?
There's no rhesus factor for the positives to attack, is there?
Good. Exactly. So a rhesus A negative blood given to an A positive person, there is no factor, there is no antibody to attack it. And even if the A positive factor, first of all, the A positive will not have antibodies because they have the factor. And when you give A negative, there is no rhesus factor, so it is safe. So negatives can be given, but positives to negative cannot. So let's look at the scenario in the case of this is very critical now. Follow me here. This is a big problem we have. Here is a woman who is pregnant. She is rhesus negative, whatever, A, B, ABO, it doesn't matter. Her partner, if you want to study the genetics of it, you go and study the genetics. What happens when a negative partner marries a negative man or the male partner or a positive? So if her partner is positive and by genetic inheritance, the baby is positive. All right, what happens here? The baby can be the mother's group, father's group. Mostly it must be the father's group. You have to look at the genetic inheritance pattern, go through all the Punnett square or whatever square, and the baby is positive. The baby keeps growing, no problem. This is the first pregnancy. There is no communication between the fetal blood and the maternal blood. There's only exchange of gases and nutrients through the placenta. Now, at the time of birth, the placenta detaches from the walls of the uterus and something happens. When it tears away, it tears the walls of the uterus, and some of the blood in the placenta, which belongs to the baby, gets absorbed into the mother's circulatory system. And remember, the baby is coming with rhesus positive blood. So what the immune system of the mother does over the next couple of weeks and months is her system recognizes there's a rhesus factor and starts making antibodies. That's fine. It stays in the mother's circulation. Baby is born, baby is fine, mother is fine, everything is okay. Then she goes into the second pregnancy. First month, no problem. Second month, no problem. Third month, big problem. Why? By 90 days, at the beginning of the second trimester, after the first trimester, the placenta is fully formed, and one of the things that crosses the placenta from the mother to the fetus are antibodies. Why? We'll talk more in the immunology lecture. The baby's immune system needs the protection before it's born, and therefore all the antibodies that are in mother's circulation is transported into the baby to protect the baby, and it's born with these antibodies. That's a protective mechanism because the baby's immune system is not going to be mature till it's between 18 months to 5 years. And you're giving birth to this baby to the environment that has all these antigens. So therefore, antibodies will cross. Now, all the antibodies are good, except that we have a problem. So assuming that she had the same partner and the baby becomes positive again, now these antibodies that were circulating in her blood goes to attack the red blood cells of the baby fetus. Two things happen in the second pregnancy. These antibodies, depending on the level of antibodies in the mother, will cause some destruction of the red cells. The baby is low birth weight. The mother goes into early labor and delivers a small for birth, early premature deliveries. This is common. All right, she gives birth to the second baby, placenta detaches, more red cells are absorbed by the mother, more antibodies are formed. So that becomes a huge problem for the third pregnancy. So now she's building up her levels of antibodies. She goes into the third pregnancy or the fourth pregnancy. Again, by the 90th day, or the third month, all the antibodies cross into the baby, and this time the load of antibody is so much it may completely destroy and kill the baby, causing stillbirths. So this is a big problem. So how do we prevent this problem? How can we save the mother and the baby? How can we save the baby? The baby is the most that is going to suffer. So here's my question. It's an exam question. Mother is rhesus negative, baby is positive, first pregnancy. Risk of erythroblastalis fetalis. That's the term used. You can read it up in your textbooks. The risk of destroying red cells is high, low, or no risk. First pregnancy, what's the answer? High, low, or no risk?
No risk.
No risk.
Second pregnancy, mother negative, baby positive, low risk, high risk, no risk.
Low risk.
Low
No
risk.
risk. Third and subsequent pregnancies, low, high, or no?
High.
High risk. What is the basis of the high risk in the third and subsequent pregnancy? The reason is at each birth, assuming the baby was positive, a few of the baby cells have been entering the mother's circulation, and the mother's immune system is developing antibodies at such high levels that at the second trimester, these antibodies will go and attack and destroy the blood cells of the fetus because it thinks it's foreign. It doesn't know. It goes to destroy it. So what do we do? Those of you who work in maternity units, labor units, what is it that you do? How do we prevent? How can we save the mother from not producing these antibodies? The only remedy is to make sure the body's immune system does not recognize the rhesus factor. And the only way you can do that is to block the antigens of the fetal hemoglobin, of the fetal red cells. So what is it that is used? Anybody, have you heard of anything that is injected to the mother at the time of labor and after labor? What do we inject?
The protein,
What's it called?
protein plasma,
What is it called? There's a name for it.
antibodies, Rh antibodies.
What is it called? RhoGAM.
Oh, Rogam, yeah.
RhoGAM are antibodies against the rhesus factor. We collect it from mothers who are rhesus negative mothers who have been pregnant. We take their blood and kindly ask them to donate their blood. When they donate the blood, we harvest the antibodies and we keep it, and we inject it into either the same mother or other mothers who are negative. What does RhoGAM do? It's a ready-made antibody that is injected. So as soon as the fetal red cells come into the mother's body, these antibodies will attack and destroy them and prevent the mother's immune system from recognizing them. Why? Because the recognition of foreign protein takes 5 to 7 days. So you want to get rid of it, destroy them before ever the rhesus antigen is recognized. So this is why we inject RhoGAM at the time of labor of all negative women or immediately after birth. But what is the problem with RhoGAM or antibodies in general injected? Antibodies injected are proteins. They have a limited lifespan of 6 months. So this woman, after 6 months, goes into the third pregnancy or the second pregnancy, is not protected. So RhoGAM is gone, dissolved and gone. They deteriorate, they are proteins, they break apart. And therefore, at every pregnancy, at every delivery, this rhesus negative woman must be given RhoGAM, and RhoGAM neutralizes the fetal rhesus positive cells that may accidentally come in. And that is the essence and the significance of knowing rhesus positive and rhesus negative status of